Yeah, so I'd like to thank the organisers. It's a great pleasure and a privilege to be here at this very inspiring location and to meet some old colleagues. Well, I shouldn't say old colleagues, colleagues I've known for a long time, and also some new people. So today I know we've had a couple of talks on phylogenetics, but I thought I would, given this audience, try to provide a little bit of a survey of some of the ways that stochastic models are used in phylogenetics. So it'll be at a fairly high level, but I will try and include some recent results that are perhaps of interest. I had somebody ask me the other day, are there actually any phylogenetic methods or any stochastic methods and models used in phylogenetics? And I can assure you there are now. In the old days, people used to analyse biological data and build trees using very sort of deterministic methods like maximum parsimony and so forth. And it was really only because of statistical approaches pioneered by people like Joseph Felsenstein, Elizabeth Thompson, and others that have led to the realisation that these deterministic methods are not, well, they're not even statistically consistent under simple models, but they're also not particularly good methods. They can, on real data, they can give very misleading answers. So that's the sort of backstory to this talk. So these are some of the processes I'm going to talk about in going from, well, of course, if you believe evolution, you believe life can be traced back to a common ancestor, and species evolved due to a process of speciation and extinction. Amaury Lambert talked a little bit about some of the models that are used in that story. Of course, these date back to Aldini Yule in the 1920s, very classical models of birth, death, Markovian branching processes. I'm not going to talk so much about those because what we're interested in phylogenetics is often taking the data we see today, the DNA sequences from genes that we obtain from different species, and estimating a phylogenetic tree for that gene. And there's this random process of site substitution that we need to deal with. So that's the stochastic process. This is the inference going backwards. But these genes, of course, might not be all of the genes. Often genes are lost and so forth. So there's another process, stochastic process at play as well. The fact that genes sometimes are not present in species. They're sampled, or they disappear, or the species go extinct. And these gene trees are not necessarily a correct reflection of the species phylogeny because of processes, both of which we've heard a little bit about at this conference. One is incomplete lineage sorting, and the other is lateral, or sometimes called horizontal gene transfer. They're exactly the same thing. This process, of course, is very important in bacteria and other, basically prokaryotes. So a little bit of background to trees, phylogenies. For us, there's some important distinctions to be made. This is what's called a rooted phylogenetic tree, and this is what you get if you ignore the location of that root and think of it as an unrooted tree. So this is actually the correct phylogenetic tree for these species. My apologies to the vegetarians. And this is a binary tree in that every vertex has two outgoing arcs, but you can also imagine non-binary trees where you might have what's called a polytomy, a vertex of out degree higher than 2, which to most biologists is interpreted as what's called a soft polytomy, meaning we don't really know what happened here with the order of speciation. Okay, there's also the distinction between trees and tree shapes. These are trees, but if I was to ignore the labels at the leaves, I would just get a shape of a tree. And finally, something we'll make use of in this talk, trees can be encoded either by knowing what all of their clusters are in the rooted case or splits in the unrooted. What does that mean? It means if I knew what was a descendant of every vertex of that tree, so the descendant of this vertex here is cat and mushroom, if I knew that was a cluster, cat and mushroom, and if I knew that daisy, rice, mushroom and cat were a cluster, if I knew all the clusters of any tree, I can uniquely reconstruct it with probability 1. There's no stochastic process here. And similarly, if I knew all of the induced subtrees of size 3 in the rooted case and of size 4 in the unrooted case, I can uniquely determine the tree. So if I knew cat and mushroom were sister taxa to daisy, so when I look at those 3 species, cat, mushroom and daisy, and I trace back the history of those 3 species in that tree, so cat and mushroom come together first, and then we have daisy. So we call this the induced subtree of that tree by those 3 species. Okay, so if I started with my original tree and I take some subset of leaves, this would be the induced subtree. So it turns out it's a combinatorial fact, not too hard to prove, that if you've got a binary tree and you know all of the induced subtrees for subsets of size 3, you can uniquely recover the tree. For unrooted trees, you need all of the induced subtrees of size 4. In actual fact, you don't need all of them. That's an interesting combinatorial story, but I don't have time to go into that today because this is not a combinatorics conference. Okay, so now let's look at these 2 processes here. I'm not going to say, as I mentioned, much about this first one, but there is something they have in common. And I'll describe that in this next slide here. Here are 3 processes that you may be familiar with, and Murray talked about some of these the other day. First of all, you're all familiar with the standard Kingman coalescent. It generates a tree, but if we ignore the order in which the coalescences occur and the branch lengths of that tree, we get a certain probability distribution on phylogenetic trees, leaf-labelled trees. Similarly, if I take a pure birth Yule tree and ignore the branch lengths, I get a distribution on trees. More generally, if I take a birth-death tree and look at what's sometimes called the reconstructed or perhaps reduced tree, what does that mean? So here's my common ancestor. It's gone through time, it's speciated. We've come across at the present and said, what do we see? Well, this is what we see. We don't see these things that have gone extinct, and we just trace back to here. Now, these models can allow the rates of speciation and extinction to depend on time. They can also allow the rate of extinction to depend on the age of a species. In that very general setting, you have these 3 different models, quite different. Kingman coalescent somehow going backward in time, these 2 going forward in time. It turns out that all 3 give rise to exactly the same discrete probability distribution on this set of rooted binary phylogenetic trees. I should stress that the set of rooted binary phylogenetic trees on N labelled leaves is finite. The number of such trees is the product of the first 2n minus 3 odd numbers. Okay, so 1 times 3 times 5 and so forth. This equivalence has been pointed out by various people and proved more formally in a recent paper. And we're going to refer to it as the Yule Harding distribution. So the fact that these 3 distributions are the same is very helpful because it allows us to prove things about, well, even about the shape of Kingman trees by thinking, well, if we ignore the branch lengths, I may as well, it may be easier to work with Yule trees, for example. Now, Amaury Lambert pointed out these branch lengths in these 2 types of trees are very different. Coalescent trees, you have these very short things at the present. Yule trees, you don't. But you have to bear in mind here we're ignoring branch lengths. So a basic property of this distribution concerns, if I generate a tree and I want to know how many are on the left of the root and how many are on the right of the root. There's nothing particular about left and right here, there's no sort of planar embedding, but I'll just choose one of them at random and call it left and one call it right. So let's think about Yule pure birth, because that's equivalent to these others. So we start with two lineages, we choose one of them uniformly at random and we make it split. Okay, so this one here we've got two on the left, here we've got two on the right. Now we've got three lineages, we choose one of them uniformly at random and make it split. So I could split that leaf or that leaf, so I get those two shapes. I could split this leaf, but if I was over here I could split that leaf and get the same tree. So what you see is having three on the left and one on the right has probability 1/3. There's two of those 6 ways will give me that, and a 1/3 probability of an equal split, and 1/3 like this. So what's going on here? I think the nicest way to think of this is this process, although we're dealing with trees, is nothing more, if we're thinking about the number on the left and the right, as a simple polyurn model, where we start with one red and one blue ball, and we choose one of the, red is of course left and blue is right, we choose one uniformly at random, just replace it with a ball of the same colour, okay, and so forth. Now, it's been known for a very long time what the distribution of red and white balls in the standard polyurn model is. It's the uniform distribution. And this turns out to be enormously useful for proving things about the shapes of Yule or coalescent trees. I've seen many complicated arguments of results that can be proved much more easily using this idea. Okay, let's look at, we're going to use that observation in our first little story. I'm going to talk about all of these, but only about 4 or 5 slides. So let's look at incomplete lineage sorting. So we've heard about this already. You've got a species tree. These are the fat tubes, if you like, and you've sampled a gene from this individual, and this individual, and you're tracing back their ancestry of that gene. This person got that from one of their parents and so forth. Now it could be that these lineages coalesce somewhere in here, and then this lineage coalesces. That would be nice, because in that case the gene tree, which was this topology here, agrees with the species tree. However, it's possible that there's no coalescence in this period, and we go all the way back to the ancestral population, in which case we could have B and C coalescing first. If we go right back here, each of these 3 lineages, assuming the population's well mixed, back here is equally likely to be the first to combine. And as soon as the first combines, that determines the tree. And now we get a different topology. This gene tree would say B and C are more closely related to each other than they are to A. So, of course, it depends a lot on the length of this period in here. Length here is measured in, I guess, time divided by effective population size, because within a single branch of this tree, we've just got the standard Kingman coalescence. So this length is being measured in in coalescent time units, I guess. And from this little argument here, the probability your gene tree matches the species tree, if it's coalesced in here, well, it'll be 1, and that's the probability coalescence in there. If it doesn't coalesce in there, which would be e to the minus L, then there's a 1/3 probability. Each of the 3 topologies is equally likely because the coalescences happen back here. And similarly for the other 2 trees are the same. So I remember when I was doing my PhD, I won't tell you how long ago that was very long ago, but people were still arguing a lot about the relationship between human, chimp, gorilla. And the reason was, one group would sequence a gene and they say, human and chimp are closer than they are to gorilla. and another group would sequence another gene, and they say, oh, no, that's wrong. No, we've got actually 2 genes, and they say chimp and gorilla are closer. And so there's vigorous fights in the systematic community. But of course now you can sequence thousands of genes. And this model makes very nice predictions. It predicts, this is bigger, and the second 2 should be about the same frequency. Moreover, the degree to which this is bigger than this is going to tell us something about this length. In other words, the combination of time, the time of separation, and the effective population size. So if you know one of those 2 parameters, you might be able to estimate the other. So there was a nice study done a while ago now, but this sort of prediction here of the second 2 trees being roughly equally supported is borne out there. And these authors were able to get some estimate of constraints on the time of separation. So it's a nice example of how, this is a very simple, there's nothing deep or difficult about what I've talked about so far. A very nice example, I think, perhaps for students of how simple probabilistic ideas can be helpful for biology. Okay, what about when we go to n equals 4? Now, we're going to do a little thought experiment. We've got our 4, some sort of a species tree. Maybe it looks a little like this. And what we're going to do is we're going to let the branch lengths of this tree be really, really short, these lengths here. So we're going to force all of the coalescences to happen before the root. We can do that, okay? We'll just make these really, really short. So it's going to look something like, okay, so everything is, all the action is going to be down here. Right, so what sort of a tree am I going to see down here? Well, we saw in the beginning of my talk, using the polyurn idea, the probability we get 1/2 or 2 leaves on each side is 1/3. The probability we get 3 leaves on one side and 1 on the other is 2/3. Okay, so there's a 1/3 probability of getting this tree shape, but we're not interested in the shape, we're interested in the tree. So we have to ask, how many ways can I label the leaves of that tree? Well, there's 3. I can have, if the 4 leaves are A, B, C, D, I can have A, B, C, D, A, C, B, D, or A, D, B, C. So that's why I have to multiply by 1/3. What about this one? Well, as I said, probability of that shape is 2/3 by that polyurn argument. This tree, there's 12 ways to label it because, well, let's see, I've got 4 choices for what I put here, then I've got 3 choices for what I put there, and then everything else is determined. I've got some symmetry here. It doesn't matter if it's CD or DC, there's symmetries in these trees. So what do you notice about this? First of all, the probability of this shape is twice the probability of that shape. However, the probability of this tree is half the probability of that tree. So what that means is if I had started off with a species tree that looks like this, and I do all the coalescences down here, the probability that my gene tree matches that species tree will be 1/18. The probability of getting any of the other 3 trees of this shape will be 1/9. Okay, it'll be twice as likely. So that shows us that it's possible, and of course these don't have to be 0, they can just be small. It's very possible, therefore, for us, for the most probable gene tree to be different from the species tree. And that's a real problem because it means if you're estimating the species tree by doing some sort of a vote, which is the most popular, or the most highly represented gene tree, you're going to get into trouble in a situation like this. You'll be estimating the wrong tree. So you notice in this example, we started with an unbalanced tree, a caterpillar, and we got support for a more balanced tree. So maybe that's a sort of a general phenomenon. Is that true? Well, we have to go beyond 4 to understand that. We say that a gene tree is an anomalous gene tree for a species tree if you can assign some branch lengths to that species tree so that the probability this randomly generated gene tree, it's generated by this incomplete lineage sorting model, the probability that that matches this tree T, if you're generating on the species tree with those branch lengths, is bigger than the probability the gene tree is the same as the species tree. So we just exhibited an example of an anomalous gene tree in the case n equals 4. It was just take any labeling of this tree, something like A, B, C, D, and if my species tree TS was this, then this would have been an anomalous gene tree. So in a very important paper, Noah Rosenberg and colleague James Degnan showed that when you get to 5, in fact, it doesn't matter about the shape of the species tree. It could have been balanced, or very balanced, and you can still get anomalous gene trees. I was going to go through the argument, but I went through my slides last night, and all I really did was threw stuff out because I realised I had far too much stuff. So if you're interested, I can explain that later. Yeah, so any simple voting scheme is going to get into trouble. However, statistically consistent methods for estimating the species tree from the gene tree given independently generated gene trees under this lineage sorting model do exist. And there's been a lot of interesting work by people using probabilistic and combinatorial techniques to construct these, and these are used by biologists now. I'm not going to describe those, I'm just going to give you a simple proof that such consistent methods exist. So, as I mentioned at the beginning of my talk, every rooted binary tree is uniquely determined by its induced rooted triples. Okay, if I take each triple of leaves and look at the induced tree, if I know all of those, I know the tree. And as we saw above, when we've just got a 3-leaf tree, we never get this inconsistency. The best supported gene tree is always the same as the species tree. Therefore, putting those two things together in a short argument, you can conclude that one consistent way to reconstruct a species tree is just to reconstruct for every triple using a majority rule vote what the most probable, what the most well-supported gene tree is, and then combine those into a species tree. That is not the best way to do it, but it's the simplest way I know that can be explained in 3 lines without any equations. So.
Any combination of the 3-leaf tree will be consistent?
Eventually, for long enough. We're talking about as consistency here in the limit as the number of independently sampled gene trees goes to infinity. Yeah.
Do you mean tau here, right?
Sorry?
So do you mean does tau can do, or is it capital T? There's twice.
I guess I mean TS, the species tree. Yeah. Mm-hmm. So what about clusters? You can actually reconstruct also remember the clusters were the subsets of leaves that were descended from vertices. There's been work on those as well. If you look at the expected frequency of gene trees that contain a cluster, one nice result due to Elizabeth Orman and colleagues is any cluster that's in basically more than a third of the gene trees will necessarily be in the species tree. Okay, the last thing on this story is what are called wicked forests. So what is, actually I think I might skip this because I'm still going to have to throw things out, so let me, I just like the title of this paper, There Are No Caterpillars in a Wicked Forest. I think it's delightful. Okay, right, lateral gene transfer. So there's this idea that amongst certain domains of life, particularly prokaryotes, that trees not particularly useful. There's been so much lateral gene transfer. There's estimates by Bill Martin, for example, that every gene in bacteria has been exchanged on average about once in its history between lineages of a tree. There's another view, though, that's perhaps become a little more prominent by people like Eugene Koonin, that there is still, even in the prokaryotic world, a central tendency statistical tree, if you like. and that these transfers sort of are an overlay on that tree. So you can model lateral gene transfer in various ways. You might have, for example, a simple model where there's a constant rate at which a gene is transferred from any point in a tree to any other point at a constant rate lambda. So there's a rate at which for any point you send out an arrow across, it hits something uniformly at random that's present at that time. And if this gene is coming down here, it goes across and the copy of the gene that's coming down is replaced by what's over here. So if I had, so I might have A and that gene is present there, this gene's coming down, but now the copy of it, if I was to trace it back in time, it comes up here and follows this arrow backwards. So now the tree is looking like, for that gene, is looking like this. It's come up and joined with that. Is that clear? I'm running a little short of time, so I'm trying to get the idea across quickly. So yeah, the total number of transfers in the tree, then of course, if you've got a uniform constant rate model, will be Poisson, and the expected value will be lambda times the total length of the tree. This is the PD of the phylogenetic diversity of the entire tree that Murray talked about. Let's think about the simplest case of 3 leaves. 3 is always pretty easy. Now you can imagine there could be, you know, we don't put any restriction on how many transfers there are. It could be a very complicated story here, but it's actually very simple because when n is 3, all that actually matters is the first transfer that happens when you go back in time. You see, if the first transfer that happened, say, was there's nothing after that. What that means is that B is now attached, sorry, B and C are now attached, and they can never be unattached by any earlier transfers. And over here, if this was the first transfer going backwards in time from the present, now A is attached to B, and nothing that happens earlier can matter. So this is very helpful because you see we have for the first transfer that occurs, there's 6 possibilities. 2 of them will be between here, 2 of them will be between here, and 2 of them will be between the A and the C lineage. So each of them will give rise to a different tree. It could, on the other hand, be that the first transfer occurs above this point. Now, if that happens, we necessarily have the original tree topology. So that allows us to write a very simple equation for the probability that the lateral gene transfer tree agrees with the species tree. It's 1 if the first transfer is up here. This P is the probability that the first transfer event happens after this vertex here, right? So you see what happens, the true species tree is the most likely LGT tree, a little bit like what happens in the lineage sorting story. But things get more complicated when you include additional lineages. Let's look at this picture. Here, imagine these are, for simplicity, the only 2 transfer events. I'm not interested in this lineage, I'm only interested in the relationship between A, B, and C. As I go back in time, B comes over here. Now, if there was no other transfer, what would happen is B would then coalesce with A, and we would have the tree A, B, C. However, what this second transfer does, if we have this transfer and then we have this transfer, well, now that combines C with B. So we have A, B, C. We have a different tree. So this is in stark contrast to what happened in the previous slide. Previous slide, all that mattered was the first transfer. Now, things can get moved, and at some point they can get fixed. So we have this distinction between moving and fixing transfers. Okay. Now this is also in contrast to what happens in the incomplete lineage story, incomplete lineage sorting story, because in that situation, the presence of other lineages has no effect. you can ignore them. But here these other lineages can play a role because you can have transfers in and out of them. Okay, so you can't...
Could you explain what you mean by moving versus fixing?
Yeah, sure. So if sigma 2 had not been there, sigma 1 would have been a fixing transfer because it would have determined that topology, and even if there were other transfers up here, it would have still been fixing. It's just that, yeah. So to analyze this model using stochastic techniques, the hard part really is just to figure out what is the process of moving transfers before the first fixing transfer occurs. And it turns out rather nicely that this question comes down ultimately to looking at a random walk, a simple random walk, continuous time where the probability of going left or right is the same at each point. So you've got 6 people sitting around a table and they pass the bottle of port to the left or the right with equal probability. And we started up here, and so we want to know what is the probability after a certain period of time, and that time is random because it's the time till the first fixing event, that we're either here or we're at this other case, which also has the same tree topology, A versus BC. So you can actually, when you do this, you find something rather interesting. You find that in a certain region, the matching gene tree topology now can be less than the other two topologies. So the most likely gene tree, the least likely gene tree can have the same topology as the species tree. So it was a little surprise, and it was also, again, in stark contrast to what happens in the incomplete lineage sorting story. So we were able to try and estimate, despite this inconsistency, you can still, as long as the lateral gene transfer rate is not too high and you have enough genes, you can still infer the species tree in a statistically consistent way. The amount of we looked at Yule trees, and we showed that, this is also Sebastian Rock and Sagi Snare, we showed that as long as the expected number of gene transfers was basically in the order of n over log n, you could infer the species tree consistently if you had enough gene trees. We did an actual instance here using our algorithm where we showed that using this formula actually, if you had 200 leaves and suppose each leaf is transferred on average 10 times, and that's very high. I mean, remember Bill Martin says each gene's transferred on average 1, which caused a lot of concern. Then it's still possible if you have enough gene trees to recover that species tree. We did some analysis with some biologists. There's this group of bacteria called Actinobacteria that are estimated to have extremely high LGT rates, and we were able to reconstruct trees that somehow broadly agreed with their analysis. Okay, how am I doing for time?
We still have 15 minutes.
Okay, great. The next thing I want to look at is what happens when all of the genes are not present. This is a problem that Michael Sanderson and his colleagues raised with me as an increasingly important in this era of genomics, that you have genes that are sort of present in some species, denoted by an X here and absent in others. Metazoa, 55% of the matrix of presence and absence is in absence, and in some of these other groups it's very high, 96%. So what are the implications of that for phylogenetic tree reconstruction? So we need this notion of what we call decisiveness. So we've got a phylogenetic tree T and a leaf set X. We're going to say a collection of subsets of X with union X is decisive for a tree if T is the only tree that displays those induced subtrees. And as I said at the beginning, if you know all of the subsets of size 3 or 4, you can reconstruct the tree. So that would be decisive coverage. And we say S is phylogenetically decisive if it's decisive for every phylogenetic X tree. So here's an example. This pattern of taxon coverage is decisive for 6 trees like this, including this tree. If I know what this does, this tree, how it restricts to A, B, C, D, that would be the tree A, B, and then C, D, and similarly for the other locus, I could only put those two trees together in one way. But there's 9 trees like this where those two induced subtrees don't determine it. For example, A, B, C, and E. I could put E over there with C. It would still give me the same induced quartets as this tree does on those two loci. Here's an example of a pattern of taxon coverage that is phylogenetically decisive. And of course, the natural question for somebody interested in combinatorics was, can we characterize when is a pattern of taxon coverage decisive? And we were able to do that. I'm not going to explain this theorem. The problem with this theorem is that it doesn't lead to a polynomial time algorithm, and we still don't know whether there is a polynomial time algorithm. It's a very good combinatorial question, but my money would be on that being hard. But I'm going to concentrate more on the random process part of the story, which is, imagine your pattern of taxon coverage is just determined by the toss of a coin where the probability of heads is P. If it's heads, you're going to sample that taxon. So you've got a tree with n leaves, you've got a probability P of sampling X taxon, and you're going to generate K data sets. So the question is, what is the probability that that collection of data sets, subsets of X, is decisive for that tree, or maybe decisive for all trees? So this is basically the result. It will be decisive provided K is at least basically log n over P, P cubed, sorry. P is the probability of coverage. And to get decisive for all trees, you really just have to multiply that by 3. A little surprising, because decisive for all trees is a much stronger notion. So we've been applying this to we've got lower bounds as well. The lower bounds are remarkably close to these upper bounds, actually. Here's an example of how this would apply. Suppose you've got 100 taxa, and we expect 50% coverage. How many loci do we need to be 95% certain that they will be decisive for that tree? So it turns out that our theory gives us that 57 would suffice and 49 would be required, or may be required depending on the shape of the tree. So these are not so far apart. The last thing I'll say on this topic is we have really applied this to biological data. We've looked at how our predictions would apply to real data sets. And some of these ideas have also led on to the study of the landscape of methods like maximum likelihood. When you analyze different genes separately, we came up with this notion of what we call terraces of trees, where you can have very, very flat likelihood regions. This maximum likelihood for the gene trees, for the species tree, given these gene trees. But we explored using some combinatorial techniques. We showed, for example, if you removed 12 of these trees, 12 of the taxa from these trees, you could reduce this huge terrace of 61 million trees down to a single tree. So that was quite nice. Okay, so that's the end of that story. I've got one final story, which is on site substitution models. So the way you get from these gene trees to sequences is you look at say, a DNA sequence aligned for some particular gene, and you look at a site, and here you've had C, you've got A, so there's been some sort of mutation in the past. Maybe, for example, this was all A, and it changed to C and C at the common ancestor of cat and mushroom, for example. Right? So we're going to call this thing a site or a character. Okay, so, but you can think of it as a letter or a colour that you put at the leaves of the tree. This is C, this is A. No, what is it? This is C, the others are A. Okay, so to summarise a big story, the models that are typically used, they're very widely used in molecular systematics. They typically assume sites evolve IID according to some continuous time Markov process. there's a lot of work being done in showing that the tree is identifiable from the probability distribution on those site patterns under mild conditions. There's certain restrictions. This is a very detailed and long story, summarising it in one slide. But if you assume a molecular clock, that is the rate of substitution is constant throughout the tree, it turns out there's a very easy proof in many cases that you have statistically consistent ways of recovering the tree. And unfortunately, several authors have missed this simple observation and gone into very complicated arguments that you can reconstruct a tree under a certain model with a molecular clock, not realising that it can be done very easily. Okay, so the question now is, how much time do I have? So 10 minutes, fine, great. That'll easy. So the question now is, how much information is there in these sequences? See, as you shrink an edge in a tree down, you're going to need more and more sites to sort of see that edge, to see that that edge was there, because you've somehow got to have a mutation across there. And a mutation across there could look the same as two mutations on either side of it. So, and on the other hand, if you've got very long edges, then as a taxon gets very far away, you may have had many, many changes. So you essentially have what's called site saturation. You lose the signal. So as the length of this edge grows, I've used capital T here, which was a bad choice because it looks like tree, but it's the length of that edge. The sequence length you need to reconstruct this tree grows exponentially with T. And as T shrinks, for the infinite state model, or the Kimura infinite alleles model, K grows at the rate 1 over T. But for a finite state model, it grows at the rate 1 over T squared. And there's good reasons for that, which I can explain later. But actually, if everything's shrinking to 0, then it's at the rate 1 over T, which is important for people studying things like, I don't know, sort of, people use phylogenetic methods in looking at tumour development and so forth. Okay, so we've got these two factors. So imagine, I mean, some of the hardest problems in systematic biology concern deep divergences in the tree of life. The origin of animals, for example, around the Cambrian explosion period. So you have a short period of very rapid radiation and then a long period of time. So we know that the sequence length you need, as the length of that short radiation goes to 0, grows that fast, and we know when you've got a long period of time, you need that many sites. But how do they combine? Well, do they add? It turns out they multiply, and this is maybe a mathematical reason why there's so much, why it's so hard to resolve some of these early divergences. I think I'm about 2 or 3 more slides from the end, so that's good. So, some interesting information theoretic questions arise. How many sites are needed to accurately reconstruct a tree? So, a simple thing to do is just count data sets. If I've got k characters for n species over an r-letter alphabet, of course, the number of data sets trivially is just r to the n to the k, which is r to the nk. That's an upper bound. The number of binary trees, if I take the log of this, and play around, I can get this. Now, if I'm going to reconstruct every tree accurately, the number of data sets had better be bigger than the number of trees. Okay? Otherwise, we'd have 2 trees giving the same data set. And that tells us that sequence length we need has got to be at least log n. Grow at that rate. What's remarkable is that actually that is the precise rate at which tree reconstruction can be obtained. In the infinite alleles model, you can do this provided the probability of change on any edge of the tree is less than half. A much harder result, due to Mossel and colleagues, is this result that in a two-state symmetric model, log n is also enough in this region here. This is a very, very difficult theorem. This theorem relies on just some simple combinatorics and uses some arguments from branching theory, branching process theory. Okay, so putting the pieces together, I've dealt with these three things separately. It could be a whole nother talk about how you combine these, and this is a very active area of research. I'm just going to mention one thing, which is combining, and I should say, the thing I'm going to mention is quite controversial. Amore talked about a controversial issue in science. Here's yet another one, and this was all about, is a method statistically consistent when you try and combine this process with incomplete lineage sorting. So, and this debate is carried on. I'm happy to discuss it later, but I won't now because I'm running out of time and I'm also on video. So let me just explain the simple thing about combining ILS and site substitution. So imagine you have these different genes, and one thing you might do is you might just stick them all together and forget that they've come from different genes and think of this as a really long sequence. And just analyse that as though it's a single sequence that hasn't evolved under any incomplete lineage sorting. Now, why would you do that? Well, it's easy, and biologists do it. And is it a good thing to do? Well, it's an interesting question because, of course, it's model misspecification. If you apply, it is possible, and this is very surprising, to come up with a simple, statistically consistent estimate of the tree topology in this setting. If you apply maximum likelihood estimation, thinking of this as just a single sequence not subject to incomplete lineage sorting, this was very controversial. Is this consistent or not? People did a lot of simulations, but eventually we showed that, in fact, it is provably inconsistent. And this lies at the heart of that earlier controversy. Okay, that's it. Thank you.
Okay. I don't even know where it is. Okay. So, thank you very much for this nice overview talk, and Etienne knows how to use this. So, how many questions? Maybe start with
Thank you very much, Mike, for this wonderful talk, and sorry to have arrived late. Two questions. So the first one is, if instead of sampling, so in the question of decisiveness, if instead of sampling each gene at random, regardless of the phylogeny, you assume like a simple model where you lose genes at a certain rate, you get some signal, right, from the knowledge of which genes are present in your species. So my first question is how can you improve your bounds in this model? And the second question is, will you tell us about this simple argument that you were mentioning? And you said that there was a simple argument that what was it again?
With molecular clock?
Yeah, exactly,
sure.
Yeah.
Happy to do that. So in regard to the first question, I can't really say much because I haven't looked at that. It's probably a good thing to do. For the second, the second point about if you have a molecular clock, oftentimes all you need to do is look at the just the uncorrected Hamming distance. What proportion of sites are different in the two sequences? And now, of course, the true distances are some transformation of that, but it's a monotonic transform. And if you've got a molecular clock, these distances, the corrected ones, are ultrametric. So the two larger ones are equal and greater than third. Now, when you do a monotonic transformation, you still have that property. So models that allow arbitrary rates across sites, as long as you have a reversible time reversible continuous Markov process, you know that this distance will be monotonically decreasing with time by the sort of spectral results. Yeah.
Is it now working? Yes, it is. With your last result about the concatenation of sequences, when you said it may be inconsistent, I guess you assume that the genes evolve independently, so meaning that there is free recombination between them.
Yes,
But
yes.
they might be linked or partially linked, so
Sure.
in fact the degree of recombination might play an important part
Indeed,
in
indeed,
this.
we were totally ignoring that feature. The proof of that result is the one and only time I've ever used the Ewan sampling formula in phylogenetics, but it seemed to be important in that proof. So, yeah.
I was wondering if people have used the branch lengths or the numbers of characters in the models of lateral gene transfer also, because the way you presented it, at least with 3, you said that all that mattered was the most recent transfer event, but other ones in the tree would make the tree shorter or taller at that locus, right?
Yes, that's true. Actually, the only the last one mattering is only the case when you've got n equals 3 and no other taxa around. But yes, I think biologists I know have done things like that in a non-rigorous way to try and sort of see what effect on branch lengths it has. That's true. Yeah.
Another urgent question. Before I reach you.
One question you said, for a binary tree, knowing the triplets is enough. Is there any result like this for a multiplicating tree?
Yes. Knowing all of the induced 3 subtrees is enough. The problem there is that, with binary trees, if you know them almost exactly, or if you may not know if you've got a short branch, I mean, it's very difficult statistically when you're doing probabilistic arguments to determine between that and that. But if you know the tree is binary, you can do that. But yes, it does extend to non-binary trees. Yep.
Okay, so I think we should continue with the next talk, and thank Mike again for it.